يسعد مساكم جميعا. Today we're gonna proceed and continue what we had yesterday about chapter 12. We stopped at slide 27 about the phases of mitosis, and we said mitosis consists of 5 major phases, which are prophase, prometaphase, metaphase, anaphase, and telophase. So if we move to slide number 28, we can see schematic diagrams for all the phases, and we should know what happens in each phase. As you can see in this schematic diagram, the cell in the cell cycle, in the prophase, it stopped in the G2 of the interphase. So we can see the beginning of the formation of the centrosomes, and each one has 2 centrioles, and we're going to talk about each one of them in details and the function of each part. So from that phase, the cell enters the mitotic division, الانقسام المتساوي, and it starts with prophase. As we can see in slide 28, prophase is characteristic by the formation of the spindle fiber اللي كنت تاخدوها بالمدرسة اللي هي الخيوط المغزلية, and the nucleus starts to vanish, تختفي, and the nuclear envelope يبلش برضو يختفي. And as you can see, the chromosomes of the cell got thicker and shorter, بيقصروا وبصيروا أتخن. And as you can see in the picture, it has 2 sister chromatids, which means chromosomes already replicated or duplicated in the S phase of the interphase. Move from prophase, we can see that the centrosomes arranged in each pool of the cell, and the spindle fiber الخيوط المغزلية بلشت تتكون, and some of it is attached to the centromere of the chromosome, and some of them to non-centrometric part. Move on to slide number 29. metaphase, الطور الاستوائي. من اسمه الطور الاستوائي, the main characteristics of this phase that all chromosomes are lined up in the middle of the cell. زي ما احنا شايفين هون الآن كل كروموسومات صارت في وسط الخلية, and all attached to the microtubules of the spindle fiber. الآن بننتقل لطور الانافيز اللي هو الانفصالي, and it's called anaphase أو انفصالي لأنه الآن الخيوط المغزلية راح يصير لها depolymerization, راح تنكمش, and it pulls each chromatid towards the pool of the cell. فهي راح تشد chromatid. احنا نتذكر الآن من الدبليكيشن صار عندنا كروموسوم أو duplicated chromosome with 2 sister chromatids, يعني في عندنا 2 chromatids. الآن each spindle fiber راح يشد each chromatid toward the pool of the cell, and in telophase and cytokinesis راح يبلش عندي بالتيلوفيز راح يبلش عندي بداية الcytokinesis, and we ended up with 2 cells. Each one of them is genetically identical to the mother cell, الخلية اللي بدأنا فيها, والأهم من ذلك إنه these 2 cells have exactly the same number of chromosomes as the mother cell. لهيك بسموه الانقسام المتساوي. الآن ننتقل لسلايد 30 و 31 و 32 هم عبارة عن إعادة للي حكيناه بس بصورة مكبرة. سلايد 33 و34 وهي عبارة عن مايكروسكوبيك ايمج محطوط عليها سودو كولرز ملونة بألوان بتبين لي شو اللي بصير بكل مرحلة. فاحنا راح ننتقل الآن من هاي السلايدات حتى نوصل لسلايد رقم 44. فخلينا نحكي الآن بالتفصيل شو اللي بصير بالضبط بالانقسام المتساوي. The mitotic spindle, a closer look. The mitotic spindle is a structure made of microtubules. فهو تركيب الخيوط المغزلية هو الميكروتوبيولز. And if you remember from chapter 7 when we talked about the cytoskeleton of the cell, one of the main components of the cytoskeleton was microtubules. إذا بتتذكروا كانت مكونة من التubulin. They are the thickest. شو وظيفتهم؟ As we said in chapter 7 and we repeat this again, microtubules controls the chromosome movement during mitosis. Going back to the previous slides حكينا بالانافيز microtubules أو السبندل فايبر depolymerize فهي بصير لها عملية عدم بلمرة فبتنكمش فبتشد معها الكروماتيد نحو قطبي الخلية. In animal cells, assembly of spindle microtubules begins in the centrosome. فالبداية تبعت المايكروتوبيولز بتكون في جزء يسمى centrosome و make sure you are not mix it with centromere. السنتروسوم اللي هو عبارة عن the microtubule organizing center. So for instance, if they ask you in the exam, the microtubule organizing center, it's centrosome, not centromere. The centrosome replicates during interphase اللي هو mainly بيكون بالG1 forming two centrosomes that migrate to opposite ends of the cell during prophase and prometaphase. In slide 45 there is something called an aster, which is a radial array of short microtubules. So if you go back to the images in slide 29, you can see short hairy stuff coming out from the centrosome. These are called the aster, extends from each centrosome. فهي بتكون toward the edge of the pool, not toward the chromosomes. The spindle complex includes the centrosome اللي هو حكينا اللي هو microtubule organizing center, the spindle microtubules أو spindle fiber اللي هي مكونة من microtubules and the aster. فلو بنعملهم على mathematic equation نحكي the spindle complex equals centrosome plus the spindle microtubule plus the aster. Move on to slide 46. During prometaphase, so this is we already talked about looking at the picture, but now it's written here. During prometaphase, so it's the phase between prophase and metaphase, some spindle microtubules attach to the kinetochore of the chromosome and begin to move the chromosome. What's the meaning of kinetochores? Kinetochores are protein complexes associated with the centromere. Be aware, centromere, not centrosomes. If you remember from the previous lecture, we talked about the centromere as the waist of the chromosome اللي هو الخصر تبع الكروموسوم. At metaphase اللي هو الطور الاستوائي, the chromosomes are all lined up, and we emphasize the word all lined up. كلهم بيصطفوا at the metaphase plate اللي هو الصفيحة الاستوائية اللي هي عبارة عن plane midway between the spindles two pools اللي راح تكون في منطقة الوسط في الخلية. Slide 47 explains the meaning of what we said. As you can see, the upper picture represents the arrangement of chromosomes during metaphase, as you can see, we have 6 chromosomes in the cell, and all of them are lined up at the middle of the cell, or what we call it the metaphase plate. between parentheses حاط لك imaginary، هي عبارة resembling وسط الخلية اللي هو زي باعتبار خط الاستواء للكرة الأرضية. And as you can see at the both sides or both ends of the cell pools we have the centrosome, and you can see like short hairy array coming out from the centrosome toward or facing the pool, and this is called the aster, and the spindle microtubule or the spindle fiber connecting the chromosomes, and some of them they are connecting the chromosomes from the centromere region, so these are called kinetochore microtubules, and some of them they are attaching with the chromosomes but not in the centromere region, so these are called non-kinetochore regions, and we're going to know the function of each type later on. In slides 48 it resembles the cell. This is a real image for a cell during metaphase, and as you can see all chromosomes are lined up in the middle of the cell. Slide 49 represents the centromere area in that chromosome. This is a closer look, صورة مقربة, and as you can see kinetochore is the protein complex in the centromere region. Slide 51 it talks about the anaphase اللي هو الطور الاستوائي, and if you remember from the previous lecture we talked about the cohesins, a group of proteins that attach the two sister chromatids together, but in this phase the cohesins group are cleaved بنقطعوا أو بتقطعوا by an enzyme called separase. Separase, which means this is the enzyme that separates the two sister chromatids from each other, facilitating the microtubules to pull each chromatid toward the pool of the cell. Sister chromatids separates based on the action of separase enzyme and move along the kinetochore microtubules toward opposite ends of the cell. فبصير عملية شد أو انكماش للخيوط المغزلية، فبتجذب الكروماتيدات نحو قطبي الخلية، فبصير كروماتيد على كل جهة. The microtubules shorten by depolymerizing at the kinetochore ends. فبصير لها عملية shortening. That's why it collapses and pulls away the chromatid toward the pool of the cell. Move on to slides 52. Results of a clever experiments suggest that motor proteins on kinetochore. ففي عندي أنا motor proteins, and by saying motor proteins, so this mechanism needs energy in the kinetochore region اللي موجود في السنترومير. Walk the chromosomes along the microtubule during anaphase. The depolymerization of the microtubules at the kinetochore ends occur after the motor proteins have passed. فبصير عندي زي ماتور وكأنه بشد الكروموسومات باتجاه قطبي الخلية. This is called, this mechanism, عملية الشد وسحب الكروماتيدات باتجاه قطبي الخلية. This is called the Pac-Man mechanism, and if you know, Pac-Man is a computer game, so they were saying like it's kind of like a Pac-Man mechanism, like when you play Pac-Man, this is resembling the movement of chromosomes toward the pools of the cell. So slide 53, this experiments suggest that the movement of the chromosome it's gonna happen from the chromosome part or region toward the pool of the cell, not the vice versa. يعني بمعنى آخر حركة الكروموسومات وعملية الجذب والانكماش للخيوط المغزلية راح يحدث من منطقة الكروموسومات وليس من منطقة البولز الأقطاب. وباختصار عملوا تجربة بسيطة، إذا بتشوفوا بسلايد 53 they put a mark زي علامة، وراقبوا العلامة وين من حيث وين بصير الشورتنينج للمايكروتوبيولز. فوجدوا إذا بتلاحظ بالرسمة الثانية اللي على اليمين بتشوف إنه المسافة بين المارك والكروموسوم قلت، which means إنه the movement of microtubules or the depolymerization of the microtubules happens in the chromosome region, not from the pool region. والرسمة الرابعة اللي تحت برضو على ايدنا اليمين it shows the depolymerization process. فلاحظ في عندي tubulin subunits, and if you remember from chapter 7 we already talked about tubulin as the main component of microtubules. حكينا it's a dimer، كان في عندي ألفا وبيتا tubulin، فبصير لهم عملية انفكاك وتفكك، and this mechanism is called the Pac-Man mechanism. فبصير عملية مشي الكروموسوم أو جذب الكروماتيد باتجاه قطبي الخلية. Chapter 53, it's the same repetition of the experiment, and you just focus on the mark and where the depolymerization happens. Is it in the chromosome side or in the pool side? Slide 56, it talks about the non-kinetochore microtubules اللي هي عبارة عن الخيوط المغزلية اللي ما بترتبط في منطقة السنترومير، اللي ما بترتبط مع البروتين كومبلكس الموجود في السنترومير، وإنما ترتبط على الكروموسوم في منطقة غير منطقة السنترومير. Non-kinetochore microtubules from opposite pools overlap، يصير لهم overlapping بالداخل and push against each other. هاي البوشينج هاي الميكانيزم this will elongate the cell، وراح نعرف why this is important to elongate the cell، لأنه بيعمل لي بالحالة هاي بتصير عملية السيتوكينيسيس أسهل مما تكون الخلية عبارة عن دائرة. At the end of anaphase، في نهاية الطور الانفصالي، الآن أنا وزعت الكروموسومات على قطبي الخلية. Duplicate groups of chromosomes have arrived at opposite ends of the elongated cell. بعدين بتبلش عندي عملية السيتوكينيسيس اللي هي the division of the cytoplasm begins during anaphase or telophase, and the spindle eventually disassemble. تختفي الخيوط المغزلية لأنه دورها بالحالة هاي انتهى، اللي هو كان جذب الكروماتيدز بالسيستر كروماتيدز نحو قطبي الخلية. Slide 57 realistically it talks about animal cells. In animal cells, cytokinesis occurs by a process called cleavage اللي هي عبارة عن شق forming a cleavage furrow، عبارة عن زي شق أخدودي. In the plant cell, because plant cells they have cell walls, so we cannot do the same as we did in the animal cell. لازم يصير عندي ما يسمى ب cell plate formation اللي هو تكوين الصفائح الوسطى، بتكون بتتجمع على وسط الخلية وبعدين تنقسم الخلية إلى خليتين. So make sure to understand that in animal cell the cytokinesis is happening, and it's called cleavage furrow عبارة عن شق أخدودي. Meanwhile in plant cells, since they possess cell walls, we cannot do the cleavage furrow, so we have what we call the cell plate formation. Slide 58 represents both mechanisms of cytokinesis. in animal cells versus plant cells. As you can see, the left top of the slide, A, cleavage of animal cell. As you can see, the cleavage اللي هو زي تكوين شق أخدودي لقسم الخلية إلى خليتين. Meanwhile in B اللي هو the formation of the formation of cell plates in plant cells, and this plate consists of vesicles بتيجي من ال ER والجولجي apparatus. اللي تحت هي عبارة عن رسم توضيحي للعملية. ف cleavage furrow. Imagine that you are holding a balloon in your hand and you start compressing it from the middle, so you ended up with two balloons in your hand. Meanwhile, for the plant cells, since they have cell walls, we cannot do that. So in this case, a cell plate has to be formed and divide the plant cell into two identical cells having the same number of chromosomes as the mother cell. Let's move on to slide 63. Slide 63 resembling mitosis in onion cell, which is a plant cell. As you can tell, you can see the cell wall very obvious, and it shows the chromosomes arrangement in different phases starting from prophase. So you should know, if we give you a picture in the exam, you should know which phase the plant is based on the arrangement of the chromosomes starting from prophase, prometaphase, metaphase, the anaphase, and telophase. The same thing for slides 65, 66, 67, 68. So these are the same version, but they took each phase and magnify it by itself. So let's talk about slide 69. We're going to talk about a special kind of cell division. We don't call it mitosis. So this kind of division happens in prokaryotic cells. And if you remember from chapter 7, when we say prokaryotes, we are talking about bacteria and archaea. And this division is called binary fission. So binary fission اللي هو الانشطار الثنائي. So the bacterial cell will be split into two cells reproduced by a type of cell division called binary fission. In binary fission, the chromosomes replicates beginning at the origin of replication في مناطق محددة موجودة على الكروموسوم الموجود في الخلية البكتيرية، بسموها origin of replication، بداية أو أصل الريبليكيشن، فبصير replication للدي ان اي تبعها. And the two daughter chromosomes actively move apart. The plasma membrane pinches inward. بصير وكأنه عملية انبعاج داخلي. But be careful, it's not the same as cleavage furrow, because when we see the two pictures, they kind of look alike. But when you see the binary fission in prokaryotic cells, in bacteria and archaea, meanwhile mitosis happens in eukaryotic cells. Slide 70, it shows the beginning of binary fission. So at the top we have a bacterial cell. As you can see, it has the cell wall, it has the nucleoid. Since we don't have a nucleus, so the DNA will be spanning in the cell. And the first thing before the bacterial cells divide is to replicate the DNA of it. So in this case, as you can see in green color, there are certain regions in that DNA. It's called origin of replication. And move on to slide 71. As you can see, one copy of the origin is now at each end of the cell, which means the DNA of that bacteria is already replicated, preparing the cell to divide. In slide 72, as you can see, the plasma membrane of that cell pinches inward فبصير له زي انبعاج داخلي to prepare the cell to be completely divided into two cells اللي هي خطوة رقم 3 replication finishes and the pinches is finishing and later on we ended up with two genetically identical bacterial cells. Move on to slide 74. It talks about the evolution of mitosis. As we said before in chapter 5 and 7, as they suggest that prokaryotes may evolve before eukaryotic cells. So if you look at the stages of cell division starting from bacterial cells ending up to the higher complex eukaryotic cells, we can see that انه الباينري fission could be the precursor انه هو بيكون الباينري fission او الانشطار الثنائي هو بدايات الميتوسيس but it's not mitosis. Because prokaryotes evolved before eukaryotes, mitosis probably evolved from binary fission. Certain protists طلائعيات exhibit types of cell division that seems intermediate between binary fission and mitosis. So if we move to slide 75, we can see at the top far left A bacterial cell زي ما حكينا قبل شوي هي بتصير في عندها الباينري فيشن وليس مايتوسيس، بصير في عندها تضاعف للدي ان اي تبعها، بعدين بصير في عندي انبعاج في الخلية لتكون خليتين. الآن ما بين مرحلة البكتيريا ومرحلة الإيوكاريوتس في كان كائنات حية أخرى لا هي باينري فيشن ولا هي مايتوسيس. على سبيل المثال في عندي كائنات مائية زي الداينوفلاجيليتس والدايتونز and some yeast اللي هي الخميرة. فمنلاحظ بي بالداينوفلاجيليتس صار في عندي مايكروتوبيولز، هذا الاشي ما كان موجود عندي في البكتيريا. however أنا ما عندي سنتروسومز، ما عندي كونيتيكور ريجن، so it's not advanced، هي مش متطورة، لكن فصار في عندي بدايات للمايكروتوبيولز، صار في عندي nuclear envelope which means I have nucleus. في سي الدايتونز and some yeast صار في عندي بالإضافة للمايكروتوبيولز بالإضافة للنيوكليوس صار في عندي إشي بسموه كينيتيكور مايكروتوبيولز، إلى أن وصلنا إلى الموست أدفانس أورجانيزمز اللي هم الإيوكاريوتس. صار في عندي اللي شرحناهم، صار في عندي النيوكليوس، صار في عندي كينيتيكور مايكروتوبيولز، صار في عندي سنتروسومز and so forth. الآن في concept 12.3 the eukaryotic cell cycle is regulated by a molecular control system. احنا رايحين نحكي هلا ان شاء الله عن دورة حياة الخلية اللي هو the frequency of cell division varies within the type of the cell. حسب طبيعة الخلية تختلف دورة حياتها من خلية إلى أخرى. These differences results from regulation at the molecular level. برضو كمان شو اللي بيخلي دورة حياة خلية أسرع من دورة حياة خلية أخرى؟ في عندي نظام بيولوجي موجود في الخلية هو اللي بيحرك الخلية إنه تنتقل من فيز إلى فيز إلى أن تنقسم ثم تبدأ حياة أو دورة جديدة وتنقسم مرة أخرى. God forbid اللهم ابعدنا عن الكانسر. Cancer cells manage to escape the usual controls on the cell cycle. الخلايا السرطانية لا تخضع لهذا النظام، لذلك دائما بنسمي الخلايا السرطانية الخلايا المجنونة، لأنه ما في إلها ضوابط للانقسام، فهي يعني non-limited cell division. وهون إن شاء الله راح نوقف عند هذا الجزء إلى أن نستكمل إن شاء الله محاضرتنا في الجزء الثالث من شابتر 12. ويعطيكم ألف عافية، واللي عنده أي سؤال مباشرة يتوجه على الإي ليرنينج. أنا